Next up is oral presentation 16, Stamford, Connecticut, Team 2, Mission 12, Finalist Proposal.
All right. Hello, Washington. I'm John Bolanino. These are my co-principal investigators, Matthew DiTolo, Alejandro Ross, Patrick Zaleski, and Grahith Manchada. And together with our fearless teacher facilitator, Sue Dougherty, we represent Stamford, Connecticut, Stamford High. So before we get into, well, first off, our experiment demonstrates the effects of microgravity on the cultivation of Saccharomyces cerevisiae, otherwise known as baker's yeast. And so before we get into the experiment itself, we'll go into the issue with space food transportation, which is the problem we seek to solve. And the problem is it's expensive. According to a recent statistic by SpaceX, it costs roughly $9,000 to send a single pound of water up to the International Space Station. So clearly this cost prohibitiveness is going to put a damper on our ability to colonize other planets, say, or have prolonged missions and experiments on the ISS. So the sooner we figure out how to cost-effectively produce food on the ISS, the faster we can produce with our experimentation in microgravity and in space.
So essentially, we're going to test the rate of reproduction of yeast in microgravity, and from our findings, we will be able to determine if yeast is a possible alternative to these very costly and consuming supply transports. Astronauts will be able to perform missions that would last much longer durations and wouldn't have to worry about supplies as much. Hence, missions to, say, Mars could become more economically and objectively feasible.
So why? Why are we sending yeast into microgravity or the ISS? Well, for hundreds of millions of years, all life as we know it has grown, developed, and evolved under Earth's gravity, right? So, however, after years of travel to microgravity and to the ISS and the space shuttle, we realized that actually most life, that microgravity actually has changes bodily functions, right? It actually has a major effect on bodily functions. So using this from Scott Kelly's all that stuff. So given that idea, we're sending yeast up into microgravity because we wanted to see maybe yeast also changes its bodily functions given exposure to microgravity, whether that changes its growth rate, its byproduct creation, or anything that changes its efficiency as a valuable space asset.
In addition to using yeast to create food, yeast can be fermented, and then it creates a very useful byproduct of fermentation, which is ethanol. Ethanol is used in many different types of fuels, and cultivating yeast and then being able to use it to make ethanol in space would be very economically efficient and useful, because ethanol is a major component in rocket fuel. Having a production of rocket fuel in space would allow us to have missions from the International Space Station themselves or alternate fuel sources to solar panels.
Okay, and now on to our actual experiment. So what we're doing is we're using a Type 3 FME mini lab, and in the first volume we're going to have our dehydrated yeast. This dehydrated form of yeast won't grow, so we can ensure that the experiment starts in space. In our second volume, we're going to have a solution of water and glucose. Water to rehydrate the yeast, and glucose to provide food for the yeast to grow. And so, as you can see, we're going to unclamp clamp A, which separates the two volumes, and shake it together so that it gets all mixed in and the yeast can grow. 3 days later, we're going to unclamp clamp B, which holds volume 3. In volume 3, we have salt, which when we mix the salt in with the already the water and the glucose, it creates a solution that is hypertonic to the yeast cells, thus killing it through dehydration. So we can ensure that the experiment starts and stops in space, getting rid of any effect that could happen through transport up and down. And now to analyze our data. When we get all the samples back down here on Earth, we can just simply compare the mass from before and after the growth. So when we run our control experiment on the ground, we'll have a change in mass that we can measure as a percent to account for any differences in mass beforehand. And then when we get the sample from space, we can have we have our mass from before, and then we take our mass after the fact, and we could take another percentage of that. And when we have our mass change here, we could see the difference. And if we notice any kind of huge disparity, we would know the effect. We would know that microgravity has a profound effect on the growth of yeast in space. And so here's our works cited.
And finally, we'd like to thank all of our sponsors and benefactors, everyone who's helped us to get here, not least Toshiba, Nises, and Susan Dougherty, of course. Thank you, and we open ourselves up to any questions.
Thank you. Do we have any questions? State your first name and
My name's Sydney Waters, and I'm from Athens, Georgia. And I would like to know what separates your project from all the other things that have been set up into space, like other types of yeast and stuff.
Okay, so when we were looking up our project at first, we saw, okay, there's already been quite a bit like that's been done on yeast. However, a lot of the projects that we saw, or like most of them in fact, they were about using yeast as an example for humans and see how humans would interact. What we're looking at is yeast itself and the product and the properties that could be found in yeast in space, rather than using it as an analogy for how humans would react in space.
Okay, thank you.